Welcome to the lesson on translation. We finally made it. We've been talking about how our DNA has all the genetic information to create a whole complete organism, but we talked about the fact that it takes a lot of steps before that DNA is actually expressed as a physical trait. And we know that proteins help express traits. For example, the protein melanin helps create our eye color. But it's not so simple. We know our chromosomes have DNA in them, and we know that pieces of the DNA that actually code for protein are called genes. But what has to happen first is we have to take our gene and we have to make an RNA copy of it called messenger RNA. And that messenger RNA is made through the process of transcription. Now we have to take our mRNA and take it to our protein factory, the ribosome. And in the ribosome, it's going to get translated through the process of translation into protein. And this is the process we're going to look at today. So translation is the process of decoding mRNA into a polypeptide chain or protein. And ribosomes read the mRNA one codon or 3 bases at a time to construct a protein. So we take our DNA, we make an RNA copy through transcription, and what we'll look at today is how do we take this RNA and translate it into a sequence of amino acids, into a protein. Before we can get to translation, we're going to look at the different types of RNA. There's 3 types of RNA, and all of them are involved in translation. So let's review those. So the first type of RNA is rRNA, which makes up the ribosome. So ribosome, of course, is our protein factory. We know that there's free ribosomes and there's ribosomes that are attached to the endoplasmic reticulum. And the ribosome is made up of rRNA, and it's also made up of proteins. rRNA is really important because it catalyzes the formation of peptide bonds between our amino acids. So it helps these amino acids connect to each other, bind to each other, so that we can actually build a protein. The ribosome is made up of 2 parts. There's a large subunit, which is large, and there's a small subunit, which is relatively small. So that's our ribosome, and that's rRNA. The second type of RNA we talked about was mRNA, or messenger RNA. Messenger RNA is a straight line chain of nucleotides, and it's made through transcription. It's got the actual message that's inside of the gene. And remember, the mRNA is made up of codons. Codons are a sequence of 3 bases, so 3 letters, right? You want to think of each 3 letters as a word that means something, except we don't call it a word, we call it a codon. And what it means is it's the information that is going to code for an amino acid. So the first 3, the AUG, is actually something known as a start codon. AUG is our start codon, and it codes for a specific amino acid, methionine. And I'll prove it to you that this is the case. If we look at our handy-dandy little translator, I'm going to put it in AUG, methionine. So AUG, methionine, is always the beginning, the first codon during translation. It's our start codon, so it's a really special and important one. These 3 are also special, UAA, UAG, and UGA, because these are our stop codon. So right here, UAA is our stop codon. And this is going to tell the ribosome that translation is done, our protein is finished, we can stop this process. And I'll prove it to you. Let's double check UAA on our translator. So UAA. Yes, that's our stop codon. The third type of RNA is tRNA, which means transfer RNA. And transfer RNA has this clover shape, and it also has 2 attachment sites. This attachment site is our amino acid attachment site, and you can see the amino acid methionine. We just write the first 3 letters, MET, right over here. So the tRNA is actually bringing in the amino acid so that it can build the chain. The other side of the tRNA has something called an anticodon, and the anticodon is basically the opposite of the codon. So it's going to base pair with the codon. So for example, if our codon is AUG, it's going to base pair with UAC. The A is going to fit with the U, right? The U is going to combine with the A, and then the G goes with the C. So the codon and the anticodon base pair with each other. All right, so before translation, we have the DNA separating here. So some RNA polymerase is coming in here, and it's unwinding and separating the chain. And we start to build the messenger RNA over here. And that process, of course, is transcription. It's how mRNA is made. And once that's done, the mRNA goes through these little nuclear pores, and it goes into the cytoplasm, and it's going to find itself a beautiful ribosome. So now we're ready for the translation steps. All right, so the first thing that happens is initiation. This is basically the start of translation. And what happens is the two ribosome subunits, so the small subunit and the large subunit, and the tRNA, the first tRNA, which is called the initiator tRNA, they're all going to converge on this mRNA strand. So we have the large subunit coming in, we have the small subunit, this is the ribosome, of course, and we have the first tRNA coming in. And the first tRNA is called the initiator tRNA. And if you look at the initiator tRNA, it has UAC because it's UAC that goes with AUG. And we know AUG is our start codon. It's the codon for MET or methionine. All right, so that's the first thing that's happening. This is called initiation. We have our start codon red, and we have our initiator tRNA coming in with methionine. All right, so step 2. A second tRNA binds to the second codon. This is our second tRNA coming in, and it's going to bind to our second 3 letters, our second codon. And what happens next is you're going to have a peptide bond forming between our first 2 amino acids. Look, you can see the chain right there. All right, so third thing, the first tRNA is going to be released, and the ribosome moves to the next codon. So we're going to go and we're going to read the next codon, right? And our third tRNA is going to come in, and it's going to bind with our third codon. And we're going to keep going like this and making this chain longer and longer, bringing in new tRNAs carrying new amino acids. But let's continue explicitly going over the steps. So a peptide bond forms between the second and the third amino acid. That's what happens right here. The second tRNA is released, and the ribosome moves to the next codon. A fourth tRNA brings the next amino acid to be added to the chain. The process repeats and repeats and repeats, and this chain gets longer, and we keep connecting the amino acids with peptide bonds until we hit one of those stop codons. Once we hit the stop codon, in this case it's UAA, the polypeptide is released. This polypeptide is free to go, our protein is complete, and the ribosome subunits are going to separate again. And at this point, we have actually finished translation, and we have created a shiny, new, beautiful polypeptide that's ready to be used and help you express your genes. So I really hope that was helpful. Thank you so much for listening.